Chapter 1, Introduction to Parasitology, Section Relationship of Parasitology to Other Sciences. We begin with what is referred to as descriptive parasitology. Descriptive parasitology is actually pretty ancient. Some of our earliest writings we've been able to find describe different ailments people or animals would have that now with modern medicine we look back and we recognize these symptoms and these signs as parasite infections. Parasite infections that either robbed the patient or the animal of nutrients, causing them to starve, or caused weird blisters. Or there's even descriptions of what is referred to as guinea worms, the little parasitic worm that burrows into your leg and up under the fascia, and having to pull it out by wrapping it around a stick. Over the thousands of years of history that we have been recording and describing these different ailments, we didn't know what parasites were, remember, up until, a few hundred years ago. We just, figured this was due to some bad food or bad air, bad humors, or bad, feng shui or whatever. They came up with numerous different, I have here, as you see, called, I see up here, the treatments. Well, the treatments could be anything from, drinking cow urine. Yes, I have read different ailments. They would boil down cow urine to concentrate it. Or they would smear it, boil it down so it's liquid or it's more of a paste now, urea paste, and would smear that on open sores. They would go to an apothecary who would put together some form of tincture. A tincture is when you put different herbs, different things into an alcohol-based solution and allow the alcohol to solubilize compounds. And then you take that alcohol stuff and you put it on the open sore. Now, what we also know is that these different organisms that we now know of as parasites are very broad in the eukaryotic kingdom, or in the eukaryotic side of life here on planet Earth, and are spread throughout the kingdom, Animalia, kingdom Protozoa. As parasitologists, we utilize the Linnaean classification system. We go through, we group like with like based on structure, shape, where they're found, what kind of metabolic activities and things like that. Modern science, we've moved beyond the Linnaean classification system, kingdom, phylum, order, class, genus, species, subspecies, strain, all of that. And we're moving more into an area of what is referred to as phylogenetics. How are things linked together? How are they related together? You've seen it in other classes. You've seen it in textbooks, phylogenetic trees. Each phylogenetic tree is going to be different based on what criteria was used to, go through and characterize and to link together the different organisms. You can see here some of the different phylogenies can be based on DNA sequence, phenotypes, metabolic activities, how we think they evolved. So it'll be an evolutionary systematics based on evolution, generating a phylogenetic tree to see who evolved from whom. Who co-evolved? How did these things work? When it comes to parasitology, what you're going to find is we spend a lot of time on what is referred to as life cycles. Okay? Two parasites, male, female, boom, boom. Come up, fertilized eggs hatch into some form of larval stages. The larval stages will develop through different stages until they hit a sexually mature stage, which now male, female, boom, boom, there you go again. For this to occur, we're going to find that there's going to be, as you see here, definitive and what are referred to as intermediate hosts. The definitive host, as we're going to see later in this chapter, and we're going to talk about ad nauseum in the next chapters, is going to be the host that the sexual reproduction occurs in. Any host, any organism that parasite is in, that it's either larval stage or just a holding stage, those are what are referred to as intermediate. Okay? These are going to be for asexual development, asexual reproduction. One splits becomes two truly identical, whereas sexual reproduction gives you 1 and 2 come together, give you one organism that is still the same species, but hopefully genetically unique. These intermediate are going to be vectors. A snail gets infected by a larval stage. Inside that snail, the larval stage just grows, increases in size, gets nutrients. That's it. Along comes a fish, eats snail. Snail digests away, larval parasites now inside the fish. It goes on a bit of a walkabout, penetrates out the stomach into the muscle where it goes through successive larval stages, changing not only its phenotype, its appearance, but maybe some genetic alteration may also begin to occur. Certain genes turned on, other genes turned off, things like that. Snail was a vector, fish basically an intermediate. As you see right here, humans inside us, Taenia saginata, Taenia solium, depending upon whether or not the egg, the larval stage you're eating, comes from a pig or a cow. Taenia saginata, also known as tapeworms. We eat an infected cow, the meat of an infected cow, meat of an infected pig. Larval stage gets in us, develops in our gastrointestinal tract, becomes sexually mature. It turns out tapeworms are hermaphroditic. Not only can they fertilize self, but they can also exchange sperm and cross-fertilize. So that way they get some genetic variability. We pass out eggs. Somehow eggs get inside cow, pig, eggs hatch, larvae go, get in, exit out into the muscle and develop a few stages and then hibernate until the human comes along and eats, so on and so forth. Word definitive, cow or pig is the intermediate. So you have to keep in mind, as we're going through this, you're going to hear these phrases used over and over: host, vectors, definitive, intermediate, larval, adolescent. Know now, so that way in later lectures, when I say such and such was the intermediate host, you don't have this jarring shock stop as you try to remember, what's an intermediate host? Learn it now, know it later.